The article that I chose is about a protein that could reverse brain aging. This is important because it shows that reducing a single protein can reverse some aging symptoms, at least in the brain. Brain aging and memory loss are big societal challenges. If the FTL1 can be translated, it might be a path toward different therapies. This article was published in Nature Aging, a reputable, peer-reviewed journal. UCSF is a credible institution. The researchers compared young versus old animals, measuring structural, cellular, and behavioral effects. There are some limitations though. It was conducted only on mice rather than humans, and only one brain region was studied, the hippocampus. The hippocampus is this yellow thing right here. Key takeaways from this article are that the researchers, they discovered a protein called FTL1. It increases naturally in the brains of aging mice, particularly in the hippocampus, the area which is responsible for memory and learning. During the study, when scientists raised FTL1 levels in young mice, the brains started to look and act older. They formed fewer neural connections and performed worse on memory tests. But when the researchers lowered FTL1 levels in older mice, those negative effects began to reverse. The second key takeaway is that most of the research on aging focuses on slowing down the process, such as preventing loss of neurons for delaying memory decline. When the scientists reduced the amount of FTL1 in older mice, they began making connections between nerve cells. These mice started performing like much younger ones in maze and recognition tests. The third takeaway. As we age, the hippocampus tends to shrink and lose its ability to perform new synaptic connections. That's one of the main reasons why older adults may have trouble remembering recent events or learning new things. By studying FTL1 in the hippocampus, scientists linked molecular changes directly to behavioral outcomes. When the FTL1 was reduced, they saw a physical increase in healthy neurons and synapses, but also improved performance in memory tests. This tells us that brain aging is not just about wear and tear. It can be actively regulated by proteins that affect how neurons communicate. The fourth takeaway. The older mice with reduced FTL1 were placed in memory and learning tasks. They showed struggle with forgetting where certain things were, but after the treatment, the older mice performed like young ones again. This provides a model for testing future drugs or even therapies. The fifth takeaway. The research was done only on mice. Humans are far more complex. What works in a mouse's brain doesn't always translate directly to humans. If we suppress the FTL1 too much, we might interfere with other important processes. Some scientists argue that aging is multifactorial. Many processes like DNA damage, mitochondrial decline, inflammation act together. A single protein like FTL1 may be one piece, but targeting it alone may not produce durable or safe therapies. The multiple choice question I made from this article is, what happened when researchers lowered FTL1 levels in older mice? A. The mice aged faster. B. The memory and learning improved. C. They lost more neurons. Or D. Their hippocampus shrank.